Don't stop. Don't stop. Don't stop. Don't stop. Don't.
Cartilage and bone are the two specialized connective tissues discussed in this chapter. Cartilage is a smooth, firm structure containing a flexible matrix, whereas the matrix of bone is calcified, making it inflexible. As the cells of cartilage and bone secrete their respective matrices, they become entrapped in that matrix. Cartilage and bone are intimately related through their function in resisting stresses and in supporting various elements of the body. Also, during embryonic development, hyaline cartilage is elaborated first to form the template on which long bone develops. As bone is being elaborated, the cartilage is resorbed in a formation. Most of the remaining bones of the skelprocess called endochondral bone are formed by another method called intra-membranous bone formation, in which bone is formed within a membranous sheath in the absence of a cartilage template. Cartilage, cells of cartilage known as chondroblasts and chondrocytes secrete an extracellular matrix composed of glycosaminoglycans and proteoglycans reinforced by collagen and elastic fibers. During their secretory process, the chondroblasts become entrapped within the matrix they secreted and become known as chondrocytes, occupying small cavities called lacunae. Because cartilage does not possess a vascular supply, nourishment must reach these cells by diffusion through the matrix from the vascular supply located in the connective tissue, perichondrium, surrounding the cartilage. The flexible nature of cartilage and its resistance to compression enable it to absorb shock, cover the surfaces of most bony joints. Its smooth surface eliminates frictional forces during articulation. There are three types of cartilage, and they are defined by the fibers present in their matrix. Elastic cartilage resembles hyaline cartilage except for the presence of coarse elastic fibers in its matrix that impart an opaque yellowish tinge to it and a greater degree of flexibility. There are additional subtle differences between them. The chondrocytes of elastic cartilage are more numerous and larger than the chondrocytes of its hyaline counterpart, and the matrix of elastic cartilage is less abundant than the matrix of hyaline cartilage. Elastic cartilage is present in the pinna of the ear, larynx, epiglottis, and external and internal auditory tubes. Fibrocartilage has no perichondrium and resembles tendon because it is composed of thick parallel bundles of type I collagen fibers with very little matrix. Fibrocartilage matrix, composed mostly of dermatan sulfate and chondroitin sulfate, surrounds small chondrocytes lodged between these coarse collagen fiber bundles. Frequently, fibrocartilage is formed when the tensile forces placed on tendons become excessive. Fibroblasts differentiate into chondrocytes that form matrix, transforming the tendon into fibrocartilage because it is better able to resist the powerful tensile forces placed on the tendon. Fibrocartilage is present in articular discs, intervertebral discs, pubic symphysis, and at sites where tendons and ligaments attach to bone. Hyaline cartilage is located throughout the body, including the rib/sternum joints, the skeleton of the air passageways in the respiratory system, and the skeleton of the larynx and much. Although fibrocartilage is very strong, sometimes the forces on the vertebral column may be excessive, causing the intervertebral disc to herniate or to rupture. These conditions may often be very painful because the slipped disc may impinge on spinal nerves in its vicinity. Slipped and ruptured discs occur most often on the posterior aspects of the lumbar region. Histogenesis and growth of hyaline cartilage. Mesenchymal cells located in the vicinity where cartilage is to form assemble into chondrification centers, express the gene SORS, and differentiate into chondroblasts. These newly formed cells secrete cartilage matrix, which surrounds chondroblasts, trapping each in a small space known as a lacuna. These cells still possess the ability to divide. They are now known as chondrocytes. During interstitial growth of cartilage, a chondrocyte divides to form a collection of 2 or 4 cells, called isogenous groups, within the lacuna. As each cell of the isogenous group secretes matrix, the lacuna is separated into 2 or 4 compartments. The chondrocytes move farther away from each other, and the cartilage increases in size. Other mesenchymal cells surrounding the forming cartilage differentiate into fibroblasts that form a dense, Vascular connective tissue. The two-layered perichondrium. The outer fibrous layer, composed of fibrous tissue, houses fibroblasts and a rich vascular supply. The inner cellular layer possesses mitotically active chondrogenic cells that become chondroblasts. Chondroblasts of the inner layer of the perichondrium form cartilage matrix on the periphery of the cartilage, a process known as appositional growth. Appositional growth is the major method of growth of cartilage except in locations where a perichondrium is absent, example at articular joints and epiphyseal plates of long bones. Chapter Chondrogenic cells differentiate into not only chondroblasts, but also, under high oxygen tension, into bone precursors known as O5T progenitor cell 5. The growth and development of hyaline cartilage are affected by various hormones and vitamins, Table 7.2. Matrix of hyaline cartilage. Type 2 collagen is the major component of the matrix, 40% of dry weight and slight amounts of types 9, X, and 11 collagens, whereas the remainder is composed of glycoproteins, chondroitin 4 sulfate, chondroitin 6 sulfate, and heparan sulfate, proteoglycans, chondronectin, and extracellular fluid. Two specialized regions of the matrix exist: territorial matrix, immediately surrounding each lacuna. Interterritorial matrix, constituting the matrix between territorial matrices. The territorial matrix is collagen poor, but rich in chondroitin sulfates, whereas interterritorial matrix is rich in collagen, but possesses fewer proteoglycans. A narrow band of the territorial matrix, the pericellular capsule, resembles a basal lamina and is in direct contact with the chondrocytes, perhaps protecting them from physical insults. Cartilage matrix possesses abundant aggregans, large proteoglycan molecules of protein cores to which glycosaminoglycans are covalently linked. Many of these aggregan molecules bind to hyaluronic acid, forming giant, negatively charged aggregan composites that are 3 to 4 gm in length and, due to their negative charge, attract Na ions. Water molecules are attracted to this sheath of positively charged elements, forming a sheath of hydration around the aggregan composites responsible for the high water content of hyaline cartilage, approximately 80%, and permitting hyaline cartilage to resist compression. Type 2 collagen fibers embedded in this matrix not only form electrostatic bonds with the matrix, but also resist tensile forces. The adhesive glycoprotein chondronectin has binding sites to the cells of cartilage, type 2 collagen, and components of the aggregans composite, and in this manner facilitates the adhesion among the various cellular and extracellular elements of hyaline cartilage. The smoothness of this cartilage surface and its ability to resist forces of compression and tension make hyaline cartilage an ideal substance to cover the articular surfaces of long bones. Chondrocytes of hyaline cartilage that undergo hypertrophy and die leave behind a calcified matrix that results in degeneration of the cartilage. This sequence of hyaline cartilage deterioration represents the normal course of events during endochondral bone formation. However, these same events accompany the normal pattern in aging with the consequence of acute and chronic joint pain and limited mobility. Regeneration of cartilage is mostly confined to children with only limited restoration in older adults, and when it does occur, chondrogenic cells migrate from the perichondrium to the lesion. If the defect is not too large, new cartilage fills the lesion, otherwise, dense collagenous tissue is formed to fill it.